Now that we've completed glycolysis, let's move on to the next stage, which is pyruvate oxidation and then the Krebs cycle. In the presence of oxygen, pyruvate will become oxidized. Remember, if there's no oxygen present, we're going on over to fermentation. However, we'll assume oxygen is present. We've completed glycolysis, and now we'll move on to pyruvate oxidation. Pyruvate oxidation occurs inside the mitochondria when we're in eukaryotes. So inside the mitochondria is a multi-enzyme complex called pyruvate dehydrogenase, and it catalyzes all of the things that occur during pyruvate oxidation. Now, oxidation means what? Well, it's removal of electrons. So we're going to remove some electrons most certainly, because NAD is going to arrive and it'll pick up 2 electrons and 2 hydrogens and produce some NADH. Because prokaryotes don't have mitochondria, this reaction occurs right at the plasma membrane in prokaryotic cells. For each 3-carbon pyruvate, we'll see that 1 CO2 is produced. So when you consider carbons, how many carbons does that mean are left? Great, that's 2 carbons left, because the carbons need to be tracked. We can't just lose them to nowhere. So we started with 3 carbons, 1 was lost in a carbon dioxide, and now there are 2 carbons left. In addition to the 2 carbons, we have produced 1 NADH. And there are 0 ATP, so we don't have to worry about ATP in pyruvate oxidation. So pyruvate becomes what we call decarboxylated when the carbon dioxide is removed. That leaves us with an acetyl group, which combines with coenzyme A in order to form acetyl coenzyme A, which is 2 carbons. Acetyl coenzyme A, or acetyl-CoA, is what proceeds to the Krebs cycle. So pyruvate oxidation is relatively simple. If I were going to diagram this for myself, it would look much like this. I'd have a straight line. I like to track all of the carbons that were initially there in glucose. So before we start, we have 2 pyruvate, which are 3 carbons each, equals 6 carbons. At the end, when we consider carbons, we have 2 acetyl-CoA. Each of those have 2 carbons, and so I have a total of 4 carbons left. Where did the other carbons go? Well, you'll remember that we had some decarboxylation occur. So decarboxylation means that we lost carbon dioxide, but we had 2 pyruvates that went in and 2 acetyl-CoA coming out, so we lost 2 carbon dioxides. Now I've taken care of all the carbons. The next question I'm going to ask myself is, what about the electron carriers? Because we're tracking 2 pyruvates on the way in, I'm going to keep track of this with saying we had 2 NAD pluses. They came in and picked up 2 hydrogens and 2 electrons each. So now we have 2 NADHs. And what's the third thing that we need to track? ATP. Did we generate any ATP in here at all? Nope, not at all. We have 0 ATP. So everything's tracked. So at the end of pyruvate oxidation, we have 2 acetyl-CoAs for each initial glucose that went in, because glycolysis split glucose into 2 3-carbon pyruvates. And now we've taken 2 carbon dioxides away, we have 2 2-carbon acetyl coenzyme A's. These we can now take on into the Krebs cycle. The Krebs cycle is going to oxidize those last 2 carbons that we find in the acetyl group that came from pyruvate. It occurs in the matrix of the mitochondria. And there are 9 steps in 3 segments. Again, we can look at the complex Again, we can look at the complex version, and you can take a sigh of relief here to recognize that you don't have to know every single one of these intermediates. We're just going to look at the process in general. In the first segment, we'll take acetyl-CoA and add it to a 4-carbon oxaloacetate workbench. In the next section, we'll decarboxylate, that is, take carbons away in the form of carbon dioxide. And in the third section, we're simply regenerating this 4-carbon oxaloacetate molecule. So let's look at this in a bit more detail. Here is our 2-carbon acetyl-CoA. And what we're going to do is stick the 2-carbon acetyl-CoA that we obtained from pyruvate, and we're going to stick it on this workbench. And we'll call the workbench oxaloacetate, or OAA for short. And that will give us a 6-carbon molecule. Let's consider that here we have the workbench, and here we have the 2 carbons that came from acetyl-CoA. That's all we do in the first segment. In the next segment, we're going to decarboxylate. So when we take 1 carbon dioxide away, we're left with 5 carbons in our original molecule. You might think of it like this. We're left with a 4-carbon workbench and 1 of our original carbons. Now, this isn't exactly how they're arranged in the 5-carbon molecule, but for the purposes of this class, that'll suit us just fine. When we take off another carbon dioxide later in the second segment, we're now left with the 4 carbons of the workbench. However, they're not in the same form as oxaloacetate, so they need to be rearranged. In addition, in the second segment, we also produce 1 molecule of ATP for each acetyl-CoA that goes in. So in this cycle, we're tracking just 1 acetyl-CoA. How many acetyl-CoAs were there from the initial glucose? So really, we're going to be tracking 2 acetyl-CoAs in order to keep track of all the carbons. So everything in here becomes multiplied by 2. Because after all, we had 2 pyruvates that gave us 2 acetyl-CoA. Each acetyl-CoA would have given us 2 CO2s here and 2 CO2s here, as well as 2 ATPs here. In the third section of the Krebs cycle, we're simply regenerating our original 4-carbon workbench molecule called oxaloacetate. So yes, we were left with 4 carbons over here. However, they're not in the right form, so they need to be regenerated. In the process of regenerating oxaloacetate, we'll also see that a couple of other things happen. What were we missing from before? We tracked the carbons and we tracked the ATP. Ah, yes, the electron carriers. Where are our electron carriers, and are there any? Ah, yes, we do have electron carriers. We have an NADH produced over here. In fact, we have 2 of them, because for each acetyl-CoA there's 1, and there are 2 acetyl-CoAs for each glucose. Same thing here. We have another 2 NADHs per glucose. And then over here on the regeneration side, we have another NADH, which means there's 2 for each glucose. And then we have a new one, FADH2. Now, FAD is an electron carrier very similar to NAD in that it carries 2 electrons and 2 hydrogens. We'll consider it the budget version, though, of the limousine because it carries electrons a much shorter distance. So we'll say 2 FADH2. So in total, for each glucose, let's look at the summaries. We have 2 molecules of CO2 for each acetyl-CoA. but there are 2 acetyl-CoAs per glucose. So that means if we get 2 molecules of CO2 per acetyl-CoA, then we get 4 molecules of CO2 per glucose. And if we get 3 NADs becoming 3 NADH per acetyl-CoA, then we're going to get 6 NADHs per glucose. And if we get 1 FAD, which is a new type of electron carrier, turned into FADH2, then for each glucose molecule, we'll see that there are 2 FADH2s per glucose. If there's 1 ATP produced for each acetyl-CoA, now we have 2 ATPs for each glucose. And of course, we regenerated the original oxaloacetate workbench. Let's view a quick animation to see if we can clarify some of the events of the Krebs cycle.
The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide. The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule. In order to understand how the majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain.
So let's take a quick look at how we might diagram the Krebs cycle. We have a circle, and I've divided it up into 3 sections because there are 3 sections of the Krebs cycle. First, our workbench setup section. Then the section where most of the action happens, the decarboxylation section. And then the 3rd section was for regeneration of our workbench. So into this cycle, we have an acetyl-CoA. Acetyl-CoA, which is 2 carbons, joins with this 4-carbon workbench. So we'll just stick the 2 carbons of acetyl-CoA right there. Now the workbench is set up. Let's go into the decarboxylation stage, where most of the action happens. Keep in mind that this 4-carbon workbench is going to continue its way around this cycle, all the way to regeneration. So we'll just track the carbons from here on out. So twice in this decarboxylation section, we decarboxylate, which means we take a carbon dioxide, and then we take another carbon dioxide. At the same time as each of the carbon dioxides are removed, we also will be taking electrons away. After all, it's the weakening of bonds by removal of electrons that allows the carbons to float away. So in this situation, we have NAD coming in to pick up electrons and hydrogens and leave as NADH. And this happens twice, again for the other carbon dioxide. In addition, during the decarboxylation stage, we have one more component to consider, and that's an ATP. We'll generate 1 ATP. In the regeneration stage, all that we're going to see happening is rearrangement of the 4-carbon workbench. And at the same time as we rearrange the carbon molecules in there, more electrons are available to be collected. So we'll see that there's a new electron carrier, our FAD, comes in and picks up 2 electrons and 2 hydrogens and leaves as FADH2. In addition, we'll see that another NAD comes in to pick up electrons and hydrogens, carrying them away as NADH. I apologize for the sloppiness in the end of this diagram, but it seems like the more I draw on a PowerPoint slide, the less neat the words become because of the level of the computer's processor. But anyway, that summarizes all the events of the Krebs cycle. That's about all we need to know about it. We've tracked the carbons. All the original carbons from our glucose are gone. Remember that this cycle has to go round 2 times for each glucose. Each glucose generates 2 pyruvates, and those generate 2 acetyl-CoAs. So the cycle has to go round twice. So you can double the numbers of each of these things that are lost. Be sure to write a summary of it on your diagram. So let's summarize what's happened so far between glycolysis, pyruvate oxidation, and the Krebs cycle. Glucose has been oxidized into 6CO2. That is, we got rid of every single carbon of the original glucose. We've got 4 ATP, 2 that we got from glycolysis, and 2 that we got from the Krebs cycle. There are now 10 NADs that have each picked up 2 electrons and 2 protons, and 2 FADH2s have been generated from FAD as they each picked up 2 electrons and 2 protons. At this point, all this electron transfer has released 53 kcals per mole of energy by very gradual energy extraction. This energy is going to be put to use to manufacture ATP in the electron transport chain. Let's take a quick card quiz. NADH is made during the 2 carbons in acetyl-CoA are eventually used to form if an experimental drug blocks the decarboxylation reactions that convert pyruvate into acetyl-CoA. A cell treated with this drug would not be able to complete glycolysis. Is this true or false? Here are your answers. So at this point, you should have a good diagram of glycolysis, one of pyruvate oxidation, and hopefully one of the Krebs cycle coming here shortly.